Hey everyone, and welcome to week 7 in Introduction to Life Science. This week we'll be studying a different cell division from mitosis. This one's called meiosis, and organisms do this kind of division when they're getting ready to reproduce with another organism. So these are some learning goals. Explain the difference between asexual and sexual reproduction. Explain why variation among offspring is potentially advantageous. Explain the human life cycles and find commonality and differences between it and the other two kinds of life cycles. Describe what happens to chromosomes during meiosis. List differences between meiosis and mitosis. Explain how meiosis generates genetic diversity, and describe how non-disjunction inversions and translocations occur and can lead to genetic disorders. So there's a ton of resources here to help you meet these learning goals, including terms. If you don't know any of these terms, I recommend you make flashcards and study those flashcards. Got some videos here to help you understand the difference between asexual and sexual reproduction, and then to understand the differences between the 3 types of sexual life cycles. Here's some details about meiosis. First of all, comparing and contrasting it with mitosis, and then an overview of the process. And then finally, we're going to be focused on that last learning objective, which was about mistakes during meiosis, and you can get some more information about that in this video. So speaking of mistakes during meiosis, that is going to be the focus of our discussion this week. So I want to be really clear that some genetic conditions arise because you have a faulty copy of a gene. But we're not talking about those genetic conditions in this discussion. We're talking about genetic conditions that are the result of a mistake during meiosis. This could be a non-disjunction that causes a gamete to have one extra or one too few chromosomes. It could be that a bit of a chromosome gets inverted and put backwards into the chromosome. It could be that a little piece of one chromosome gets attached to another. That's called a translocation. So I want you to identify a disease that results from one of these mistakes during meiosis. Describe the symptoms of the condition and cite your sources. So please make your initial posts by Wednesday at the end of the day, and then respond to your peers by Saturday at the end of the day. To help get to know the material from Chapter 7, you're also going to complete several activities about meiosis, starting with making your own diploid dominant life cycle diagram. It should include these processes: fertilization, meiosis, and these structures: egg, sperm, adult female, adult male, cell division by mitosis, that's another example of a process, and then a zygote. So after you make the life cycle, and possibly after reviewing that video again that compared the life cycles on the content page, please answer these questions, basically comparing and contrasting the human life cycle that you made with the other 2 life cycles. Then you're going to work on another one of those Bioman simulations that's going to hopefully help you visualize and work with the process of meiosis and really get to understand some of the chromosome terminology. That can be really tricky. So we're going to focus on that, and then part 2 is going to be this meiosis interactive. You are going to make a meiosis diagram. So basically you're going to copy this into a notebook, and you are going to draw a cell with 2 homologous chromosomes before duplication, after chromosome duplication. So before duplication, you should have 1 red, 1 blue chromosome that's large, 1 red, 1 blue chromosome that's small. The different colors indicate that one came from mom and one came from dad. Then you're going to duplicate your pairs. After this, the chromosome should look like X's. So one big X that's blue, one big X that's red, one little X that's blue, one little X that's red. And then you're going to follow those duplicated chromosomes through meiosis 1, and then show us what the products of meiosis 2 will look like. All I want to see are the chromosomes. So I know there's a lot going on with the spindle, but really I just want to see that you know what's happening with the chromosomes. Finally, I am asking you to come up with a list of 6 differences between mitosis and meiosis and a list of 4 similarities. So maybe you want to use this diagram that compares the two. Maybe you want to actually print this diagram out and then write where the similarities and the differences are. There's also an example of it written out down here. So any way that makes sense to you, come up with a list of 6 differences and 6 similarities between meiosis and mitosis. This is where you'll turn in your work. So I want to see a photograph of the handmade drawing of the human life cycle and the answer to those questions, a screenshot or a copy of your Bioman Chromosome Quandary and Meiosis Interactive. You can email that to me as well through Bioman. A photograph of your meiosis diagram, and that list of similarities and differences between meiosis and mitosis. Okay, next, there's a practice quiz on the Chapter 7 content, and then there's a graded quiz. So this one's worth 10 points. It's got a 15-minute maximum. Okay, and then the last thing in the Week 7 module is your second lab report. So you did in Week 5, there was one activity in that lab where you watched me collect data from 3 cylinders. I specifically was measuring the dissolved oxygen concentration. In one of them, there was a plant. In one of them, there was a plant, and I blocked the light. And in a third one, there was just water. So I want you to write a lab report on that activity. And remember, like you see in the sample lab report, what we're looking for is a title. It should include the scientific name of the organism used. In this case, it was Elodea canadensis. That was the plant in the cylinder. It should include an abstract where you write what the purpose of the study was and the methods. So the first few minutes of that video will describe the methods. Then in words and numbers, describe the results that you got. You should look for those results in the data table that you completed and turned in. And specifically, I would recommend that you focus on the averages. So we did 3 replicates per treatment group over time, but really what's important is the average among those 3 replicates. And then finally, come up with a conclusion and create a figure. One of the questions in the Week 5 lab was about what would make a good figure for this data set. And many of you had some great answers there. I did give you some guidance on that if I thought you could use some. So check out the feedback you got on the Week 5 lab before you turn in this lab report. Okay, so that's it for Week 7. Have a great week and reach out if you need anything.